Hello, everyone. For this discussion, I have chosen lipid metabolism. Introduction. Lipids play a key role in energy metabolism. Fat is the major energy storage in most organisms, including ourselves. A mammal contains anywhere from 5% to 25% or more of its body weight as lipids. 90% of lipids are in the form of triglycerols. We as humans are fat burners. We take in carbohydrates, burn that fat, and store the fat that we do not use for later on energy that we may need after we have been conserving for quite an amount of time, such as when an animal goes into hibernation, they need to feed and fuel their body to make sure they have enough storage to continue the supply of energy even though they are sleeping through the winter. They want to make sure that they will be able to wake up healthy and ready to start a new season. Fat serves to cushion organs and thermally insulate to keep the proper temperature. We as mammals need to be at a comfortable body temperature to live comfortably and survive in these environments that we are living in. Utilization and transport of fat and cholesterol. Carboxyl carbons of fatty acids are highly oxidized, but most of the fatty acids' carbons are highly reduced methanol. Metabolic oxidation of fats consume more oxygen on a weight basis, as you'll see throughout this process. A weight versus density basis is seen quite frequently, as our bodies know the amount of weight versus the amount of density can control how much energy or how much needs to be input or output. Fat is extremely nonpolar, making it anhydrous, meaning it will not do well under certain water conditions or will settle at the surface of water conditions. Intercellular fat contains about 6 times as much potential metabolic energy on a mass basis as intercellular glycogen. And the brain is distinctive in using fatty acids as a significant energy supply. Our brain, as stated before, knows when to alert our body that we are in need of fuel or energy of any kind to continue our homeostasis status of the healthy body temperature, the correct amount of oxygen, the correct amount of water, daily things that our bodies need to survive. Transport of fat to tissue. The transport of hydrophobic triglycerols through the blood and lymph is dealt with in part by interaction of the lipids with proteins to form soluble aggregated called lipoproteins, leading to the creation of lipoproteins called microchrones. This is a transport vessel for cholesterol to be able to pass through our body. Polypeptide components of lipoproteins are known as alipoproteins, mainly found in our liver. A total of 10 major alipoproteins are found in the human lipoproteins. Lipids are lower in density compared to proteins, again, making sure our body knows the weight versus density compared to different things that are going on. Lipoprotein classification. We're going to be looking at the structure that I have on the right side of the slide here, showing the overall structure, as there may be a difference in density or weight for certain ones, but in the end, they all have a very similar structure, as well as looking at the classifications in order of density. Some are very low density proteins, some are intermediate, some are low density, some are high density, but all lipoproteins share a common structural feature, including a circular shape, inter-care hydraulic protein structure, cholesterol, and polar head groups of the phospholipids outside, as shown with our circles on the outside in the blue. Fat digestion and absorption. Triglycerols are driven from 3 primary sources: diet, biosynthesis, storage deposits in epididymites. Bile salt is essential to the digestion of lipids and their absorption into the body. They are derived from cholesterol and allow for attacks by water-soluble enzymes. As stated, many of these are hydrophobic and cannot stand to be around water, so the fat digestion and absorption of this allows them to pass through water areas or fluid areas. Transport and utilization of lipoproteins. Chylomicrons represent the form in which dietary fat is transported from the intestine to peripheral tissue, including the heart muscle and adipose tissue. Very low density lipoprotein plays a role for triglycerols, and both are hydrolyzed to glycerol and fatty acids at the inner surface of capillaries in peripheral tissue. Some of the lipoproteins, lipase, are hormone-sensitive lipase. As our hormones send triggers to our brain, as stated before, our brain has receptors that send signals throughout our body to realize when we need certain things and to make sure that our body is receiving the minimum of everything that we can supply. The LDL receptor and cholesterol homeostasis. Complex mechanisms have an evolution to maintain proper amounts and distribution of cholesterol in response to changing dietary availability. Excess LDL cholesterol accumulates in the inner arterial walls that form fatty streaks that attract white blood cells, leading to engorged fatty deposits that harden into plaque that result in the condition known as osteosclerosis. And this blocks key blood vessels, resulting in blood clots, a lack of circulation, and many other health issues. This can lead to excess fat being left around, resulting in health risks, as stated before. Now we're going to take a look at two of the scientists that began the research into looking at the transport of cholesterol from LDLs into cells, as this was a very wide-asked question in the textbook until we got to this topic. Beginning in 1972, they began researching the transport of cholesterol from LDL into cells, as stated. Studying familial hypercholesterolemia, or FH, and how cholesterol is taken into cells through the action of a specific receptors such as the LDL receptors, which is deficient in FH patients. And this revealed a new mechanism mediated endocytosis. This study earned Brown and Goldstein a Nobel Prize in 1985 for medical scientists. Mobilization of stored fat for energy generation. The capacity of stored deposits to store fat is virtually unlimited, as we've heard. They use what comes from our diet that is absorbed and then transport it into adipose tissue for storage. Catabolism begins with hydrolysis of triglycerol to yield glycerol and fatty acid. 3 lipotopic enzymes are known to participate in fat breakdown, including triglycerol lipids, adipose triglycerol lipids, and monocycle glycerol lipids. Free fatty acids, hydrosol, exit the adipocyte by passive diffusion and find their way back to blood plasma, where they become abelum. Fatty acids are released from abelum and taken by tissue and then passive diffusion, so the fatty acids can be taken into the cells. The glycerol is released into the bloodstream and taken by the liver cells, leading to the production of glucose. Thank you guys so much for your time. Now we'll review the 2 questions I have for our discussion this week. What roles do lipoproteins such as LDL, HDL, and VLDL play in lipid transportation? And how does lipid metabolism contribute to conditions like obesity? Thank you guys so much for your time.